Blood is a highly specialized fluid connective tissue consisting of cellular elements suspended in a liquid extracellular matrix, where survival depends on precise microscopic morphology. When whole blood is centrifuged, it separates into three distinct layers based on density. The uppermost, straw-colored layer makes up about 55% of the total volume. This is plasma, the non-cellular extracellular matrix of the blood. Plasma acts as a calibrated ribambamical solvent. It carries the dissolved nutrients, gases, and specific proteins required to maintain the physical integrity of the vasculature. The most abundant plasma protein is albumin. Synthesized in the liver, it acts as a primary transport carrier for fatty acids, hormones, and drugs. Albumin's critical mechanical role is maintaining plasma oncotic pressure. Inside the microvasculature, albumin molecules exert a specific osmotic force, pulling interstitial fluid back into the vessel against the outward push of blood pressure. The remaining protein volume consists largely of globulins. The liver produces alpha and beta globulins to transport lipids and metal ions, while gamma globulins are secreted by tissue-resident plasma cells to function as circulating antibodies. When the liver fails to synthesize sufficient albumin, whether from hepatic failure or severe malnutrition, conditions like kwashiorkor, that vital oncotic pressure drops. Fluid leaks into the surrounding tissues, resulting in severe systemic edema. The failure of a single liver-synthesized protein fraction systemically collapses the fluid boundaries of the entire cardiovascular system. At the very bottom of the centrifuge tube lies the hematocrit, the dense red layer composed of erythrocytes. These are terminally differentiated cells that lose their nucleus and organelles shortly before entering circulation. Normal human erythrocytes take the shape of flexible biconcave discs. As seen here, this specific geometry provides an exceptionally high surface-to-volume ratio, placing the maximum amount of hemoglobin close to the cell membrane for efficient oxygen exchange. To navigate tight capillary turns, the erythrocyte relies on a submembranous protein lattice. Peripheral proteins, primarily spectrin dimers, anchored to the membrane by ankyrin, create a meshwork that grants the cell its required elasticity. A single amino acid substitution in the hemoglobin protein destroys this flexibility. In sickle cell anemia, the mutated hemoglobin polymerizes, deforming the cell into a rigid crescent-like structure that leads to capillary blockage. Systemic tissue oxygenation is entirely dependent on the nanoscopic flexibility of the erythrocyte cytoskeleton. Between the plasma and the hematocrit sits the buffy coat. Comprising less than 1% of total blood volume, it houses the circulating immune architecture, the leukocyte family. Neutrophils are the most abundant leukocytes, making up 50 to 70% of those in circulation. They are identified by their polymorphonuclear layout, typically featuring a single nucleus divided into 3 to 5 distinct lobes, linked by thin nuclear extensions. The neutrophil cytoplasm contains two functional granule payloads. The lighter, specific granules secrete enzymes that degrade the extracellular matrix, while the larger, electron-dense azurophilic granules act as modified lysosomes to kill engulfed bacteria. Following an acute bacterial infection, these cells quickly exhaust their resources. Because they rely on anaerobic glycolysis and possess minimal cellular machinery, millions of neutrophils die by apoptosis each day, combining with digested microbial debris to form pus. Neutrophils function as disposable, localized chemical weapons factories, engineered to execute high-intensity defense in oxygen-depleted environments. Eosinophils are far less common, identifiable by a characteristic bilobed nucleus and a cytoplasm densely packed with large, intensely acidophilic specific granules. These granules contain major basic protein, a toxin specifically equipped to kill parasitic helminths. Eosinophils also modulate inflammatory responses triggered by allergies, gathering in tissues like the intestinal lining or the lungs of asthma patients. The rarest circulating granulocyte is the basophil. Its irregular, S-shaped nucleus is characteristically obscured by massive, strongly basophilic granules that stain a deep purple. Like tissue-resident mast cells, basophils express surface receptors for IgE antibodies. When exposed to a specific allergen, the cross-linking of these receptors triggers a rapid exocytosis of their granules, flooding the tissue with histamine and heparin. These rare granulocytes serve as systemic modulators, where single-cell degranulation can cascade quickly into life-threatening hypersensitivity. The remaining white blood cells belong to the agranulocyte family. These cells lack specific granules, retaining only basic, nonspecific lysosomes. Lymphocytes make up roughly a third of all circulating leukocytes. A small lymphocyte is identifiable by its intensely condensed spherical nucleus, which leaves only a microscopic thin rim of pale cytoplasm. Monocytes are the largest circulating leukocytes. Their nuclei are distinctly indented, kidney-shaped or C-shaped, with slightly lighter chromatin than that found in large lymphocytes. Monocytes function as circulating precursors. They roll along the endothelium until chemical signals prompt them to squeeze through the vessel wall and enter the tissue matrix, where they differentiate into macrophages or osteoclasts. The mononuclear phagocyte system bridges the gap between temporary circulating surveillance and permanent tissue-level immunity. Platelets are non-nucleated fragments serving as a rapid response repair system. The peripheral hyalomere houses a marginal bundle of microtubules to maintain shape, alongside an open canalicular system for rapid degranulation. The central granulomere contains clotting payloads, including alpha granules carrying fibrinogen and delta granules loaded with ADP. Platelet activation begins when surface receptors, like glycoprotein IB, bind directly to subendothelial collagen exposed by a vascular tear. Genetic defects in these receptors prevent this initial binding, resulting in severe bleeding disorders. Platelets are highly organized structural bombs, primed to detonate their biochemical payloads the moment they encounter exposed collagen. That detonation initiates the coagulation cascade. This localized chemical reaction converts plasma proteins into a three-dimensional polymer network of insoluble fibrin, casting a net that traps erythrocytes to physically plug the vessel breach. This entire response relies on the plasma. The upper fluid layer provided the exact dissolved fibrinogen and clotting factors required to build this repair mesh. Below it, the erythrocytes provide the constant respiratory capacity required for tissue survival, while the leukocytes navigate the resulting vascular architecture to defend it. Recombining these layers into whole blood reveals a single system working in synchronization. Blood functions as a fluid connective tissue, where macroscopic human survival is dictated entirely by nanoscopic precision and ultrastructural architecture. Platelets are mononucleated membrane fragments from megakaryocytes, serving as rapid-response microvascular repair. The peripheral hyalomere houses microtubule bundles and an open canalicular system for rapid degranulation. The central granulomere contains clotting payloads, alpha granules carrying fibrinogen, and delta granules loaded with ADP and serotonin. Platelet activation begins when surface receptors, like glycoprotein IB, bind directly to subendothelial collagen exposed by a vascular tear. Genetic defects in these receptors prevent this initial binding, resulting in severe bleeding disorders. Platelets are highly organized structural bombs, primed to detonate their biochemical payloads the moment they encounter exposed collagen. That detonation initiates the coagulation cascade, converting plasma proteins into a three-dimensional polymer network of insoluble fibrin that traps erythrocytes to plug the vessel breach. This entire response relies on the plasma. The upper fluid layer provided the exact dissolved fibrinogen and clotting factors required to build this repair mesh. Below it, the erythrocytes provide the constant respiratory capacity required for tissue survival, while the leukocytes navigate the resulting vascular architecture to defend it. Recombining these layers into whole blood reveals a single system working in synchronization. Blood functions as a fluid connective tissue, where macroscopic human survival is dictated entirely by nanoscopic precision and ultrastructural architecture.